Welcome to the physiology lecture bits. This video deals with the regulation of the muscle tone. As all of us know that the reflex control of the stretch reflex is the basis of the muscle tone. Regulation of the muscle tone involves basically the control of stretch reflex by the higher centers. Let us look at how the higher centers influence the stretch reflex and how this reflex control regulates the muscle tone. I am using a Sherrington Decerebrate preparation to help us understand how the higher centers have an influence on the stretch reflex. Classical Sherrington's Decerebrate preparation resulted from the mid-collicular section at the brainstem. This is a traumatic procedure which, before the advent of blood transfusion and antibiotics, did not favor the survival of the animal for more than a few hours. The operation is performed under the ether anesthesia in an experimental animal, a cat. The common carotid arteries were ligated, the parietal bone is defined, vertebral arteries are compressed as they exit from their canals. A knife or spatula is inserted through the defined hole and is directed obliquely to slice through the hemispheres along a plane immediately anterior to the tentorium cerebellum, which is bony in cat. This transection of the brainstem occurs at the mid-collicular level. Cerebral tissue rostral to the section is scooped out and the vertebral artery compression released. Packing in swabs bathed in thrombin solution limits the hemorrhage. When the animal is blown off the anesthesia, it exhibits decerebrate rigidity. The posture is the extensor hypotonia in the anti-gravity muscles of the limbs, extension of the neck as shown in the image. What causes decerebrate rigidity? Before we understand the mechanism of decerebrate rigidity, the basic knowledge of the stretch reflex arc or the myotatic reflex arc, its components and the regulation of stretch reflex by the higher centers should be understood. Let's look at gamma loop. There is an extensor muscle and a flexor muscle shown. The extensor muscle has a spindle, the central part, expanded part, and the peripheral ends. The central part acts as a receptor zone, and when this part is stretched by the stretch of the muscle, 1A efferent activity increases, alpha motor neurons are stimulated monosynaptically, the extrafusal fibers contract, and the skeletal muscle, which is stretched, contracts. This is the basic stretch reflex. Then we have the gamma motor neurons supplying the muscular ends of the spindles. The cell bodies of gamma motor neurons are situated in the anterior horn of the spinal cord, and the stimulation of the gamma motor neurons control the spindle activity. Because they are fixed at the ends by the tendon, the central part is stretched, 1A efferent activity increases, alpha motor neuron activity increases, the muscle contracts. This is the gamma loop. Gamma, 1A and alpha. Let us look at the brainstem control of gamma loop by the reticular areas. There is a large facilitatory area in pons and a smaller inhibitory area in medulla. Both these influences act on gamma motor neurons. So the brainstem centers control the gamma motor neurons acting through the facilitatory and inhibitory drives. Therefore, the gamma loop is altered by these centers. The facilitatory area by itself is active, but constantly driven by the ascending sensory tract fibers on their way to the destination and to the arousal system. Therefore, The pontine facilitatory area is constantly driven by these inputs, and its stimulation increases the gamma activity. The inhibitory area in medulla by itself is not active but has to depend on cortical and subcortical influences. We have seen the control of gamma motor neuron by the brainstem center. There are some additional controls over the alpha motor neurons supplying the flexor and extensor muscles. One such control is via the corticospinal tracts CST, acting directly on the alpha motor neuron. Another control is through the anterior lobe of the cerebellum, which via the Purkinje cell axons inhibit the fastigial nucleus, which is the deep nucleus. The fastigial nucleus normally activates the brainstem vestibular nuclei. The vestibular nuclei control the alpha motor neuron via the vestibulospinal tracts. And it is a facilitatory drive normally. That means cerebellum has an inhibitory effect on the alpha motor neuron activity and thereby the extensor muscle activity. This is seen mainly in the animals. The humans, it is slightly different. Cerebellum has more facilitatory drive effect on the muscle tone. There is an additional control of alpha motor neuron supplying the flexors by the red nucleus through the cerebrospinal tract. Cerebrospinal tract is again controlled by higher centers, cortical centers. Therefore, the cortical influences also play a major role in the control of alpha and gamma motor neurons via the brainstem centers, ultimately the extensor flexor muscle activity. So, the multiple inputs act to control the stretch reflex. Let's look at the mechanism of decerebrate rigidity. This is the level of the midcollicular section which cuts the cortical and subcortical inputs to the brainstem areas. Please note that the cerebellum is intact in this preparation. Brain centers are devoid of the cortical influences. We have seen that the inhibitory medullary area is very much dependent on cortical drive. When such an input is cut off by the inhibitory area loses its activity. There will be less inhibition on the gamma motor, more facilitation, and this excess gamma input makes the stretch reflex hyperactive.